Hi, my name is Desai Swapnil. Let's start the assignment. First is what is the non-coding DNA mystery? For decades, scientists believed that only the protein coding parts of the DNA were important. Less than 2% of the human genome. The rest, over 98%, was labeled as a junk DNA. But this raised a mystery. Why would evolution preserve so much DNA that doesn't code for proteins? What function, if any, does this non-coding majority actually serve? Bernard's data and main conclusions. Bernard, Bernardi argued that so-called junk DNA is for useless. He introduced the concepts of genomic code, second layer of information beyond the genetic code. This genomic code is not about coding protein but about shaping the structure of chromatin, the complex of DNA and protein that determine how DNA is packaged in the nucleus. He present data showing that genomes are organized into isocores, long stretch of DNA with distinct GC content. These isocores influence chromatin architecture by determining which region are tightly packed or loosely packed. The correction between GC content and chromatin structure is strong. GC rich isocores tend to be gene rich and open for transcription, while GC poor isocores are gene poor and tightly packed. His conclusion is that the non-coding genome is responsible for encoding the 3D structure and function of chromatin, essentially acting as a scaffold of the gene regulation. This insight solves the non-coding DNA mystery. It's not junk, it's architectural. My opinion is, personally I found that architectural fascinating article fascinating. It challenged the traditional protein centric view of genetic and so the DNA has multiple roles, not just a template for protein but also as a blueprint for chromatin structure. I think Bernardi's work is major step forward because it's helped explain why genome is organized the way it is, and it offer unified explanation how gene regulation, evolution and gene architecture are interconnected. It open up exciting possibility for how we understand disease related to chromatin dysfunction, including cancer and developmental disorder, and how we might target them in the futures. Thank you for listening.